Okay, so welcome again for this lab course CL 801, right? So what we are going to do today, right? So we are going to find the critical micelle concentration of the detergents using ITC. What is ITC? Isothermal Titration Calorimetry, correct, ITC. So this is what you have got the introduction about ITC in the 6th semester, right, as a part of your biophysical course. Now this particular experiment, if you see, so I will not spend too much of time on explaining the CMC, right. Now why? Because this is a continuation of your 5th semester practical, right. So what did you do there? So you had positively charged detergent which was like C-tap. You have a negatively charged detergent which was like SDS, and then you have neutral detergent which was NP80, right, NP80. Now in the 5th semester what did we do? We found the critical micelle concentration using conductometry. Yeah, we had this conductometer, so it worked very well with C-tap and SDS. Now if you could recall, for the neutral ones you failed to get any CMC value as the lines were very straight, no kinks were observed over there, right. Now the similar kind of way in 6th semester what did you do? For the same detergents you tried to do it using fluorescence, right. Now we are going to use the same detergent one more time, right, but this time we are going to use isothermal titration calorimetry, right. So what you would realize, what you couldn't do using conductometry is possible using isothermal calorimetry, isothermal titration calorimetry, ITC, right. So this in a sense completes the whole idea which we wanted to convey. Just the same experiment, finding the CMC of a detergent, but one technique might not be good enough. Conductometry for NP40 it was a disaster, no data was available, so you have to think differently, right, and parallel at the same time the whole idea is to give you an exposure to different techniques. So this is the whole philosophy for your 5th semester, 6th semester and 8th semester experiment, right. So this completes the whole idea. You start with the 5th but you end up in 8th, okay. So if you want more description about the CMC, one can look into the 5th semester video, because that is where I had already discussed what the CMC of the detergent is, how the kinks would come in and those kind of stuff, and I will put the link for that 5th semester into the description of this video as well. But what is more important for today's experiments is to know what ITC is, right. You already know theoretically what ITC is from your biophysical chemistry course, but practically how we do it. So I would make again the diagram of ITC, understanding ITC, right. Now what's isothermal? Same temperature. Iso is same, thermos is temperature, so it's isothermal, right. So it's same temperature. We are doing at same temperature what? Titration. And as you are doing some kind of titration at the same temperature, you are measuring what? The change in heat as a virtue of that titration, right. That is how the term isothermal calorimetry, titration calorimetry, right. Now how does the instrument setup looks like? Of course we need a box, right, or let us make the box later. right? You need some kind of a cell. Let's make a cell, right? So my interest, the titration of interest is protein ligand over here, right? So that means I need to measure any change in heat as and when I am going to titrate in the ligand, correct? This is what is my whole idea, but with reference to what? The buffer, right? Now in ITC, if I say, let me rub it again and write one thing. If I am looking at the reaction like protein plus ligand, I am going to get in equilibrium a protein ligand complex. Is it that simple? Right? Maybe no. So you are wanting to understand the interaction of protein and ligand, right? Are there any other interaction what you can think about? What is happening? You are doing all of these things in buffer, right? So you have protein plus buffer interaction, and then you also have ligand plus buffer interaction, right? But and all of these will contribute to your change in energy, right? So you need to subtract these protein buffer and ligand buffer interaction from the total heat change what you will get to get the protein ligand interaction. Yeah. So in short, what you will get, so H dot, this is what you will measure eventually, minus heat because of the protein buffer minus because of the heat change because of the ligand buffer, and this will result into heat of protein ligand. Make sense, right? So every titration experiment is not just one measurement, but then you also have to have these corrections, right? These corrections to be done, right? In our case, it is what? SDS dissolved in water. It's a very simple system. So we might not need these many corrections, right? So then how does my instrument look like, and how are my curves look like, and what does that curve mean to me? That is very important, right? Relevant. So if I am wanting to measure something, then I need a sophisticated calorimeter, right? That means some kind of cell I would need, like this, right? And I need another cell. Now I will call this particular cell as the reference cell, right? And this is where I will call as my titration cell, right? Now I don't want any interference from the environment, so I have to isolate it from the environment. So what kind of enclosure should I have? An adiabatic one. Adiabatic will ensure what? There is no exchange of matter and no exchange of heat, right? And this is essential, because I am looking at very tiny little bit. Yeah, we are talking in terms of like the biomolecules or the macromolecules we are talking about. So the heat changes are very small. Now you also get ITCs for the chemical reactions, like maybe HCl plus NaOH, you will get like a lot of nutrition like water and salt you will get. So those kind of ITCs, they need not be this sophisticated, because the heat energies are so high that you will be able to measure them easily, right? So what you need? I need to close this, right? So maybe I'll draw it with something else. I will enclose it. Yeah, so let's draw the border. It is some kind of an adiabatic enclosure. and of course in your textbooks you will see multiple layers, right. So just to make sure that nothing escapes, no heat, no matter escapes from this. Now herein I am going to titrate my ligand, and then I will constantly should be stirring this system. So there is a stirrer which is inbuilt, right. Why the stirrer is essential? The homogeneity, right. So the system should become homogeneous, right, because when you are going to inject the ligand, the first interaction will be at the tip of this syringe, right, but then we want to see how much of energy is released when this whatever amount of ligand you had injected mixes into the volume what you had filled in, right. So stirring is essential. Now once you stir and then you measure all of those kind of things, how do you do it exactly, right. So these are all cells which is like having a common thermocouple, which are like very sophisticated sensors which can detect even the minute temperature variations what come into picture. Those thermocouples measure this reference cell and your titration cell at the same time. Now we are doing this isothermal thing, right. So can I rub this thing off? So let's go to the graph, the kind of measurements what you do. So I have just drawn two axes. I'll tell you what to plot on what, right. So if I am saying, oh, it's gone, wait. So if I am doing measurements at isothermal, right. So I am looking at the difference between what? A reference cell and the titration cell, right. My reference cell is everything minus the ligand, right, and my titration cell has the ligand. So my reference cell ideally should be very flat, and then whatever changes happens will be by virtue of the ligand coming into the system. Okay, this is the very simple philosophy. Now I am talking of isothermal. So if I don't do any titration, this is the kind of curve which I look like. Now what exactly is on my axis over here, right? It should be like what? It is like microcal per second, and here it is the time, right, microcal per second. Now my microcal per second is what? Power, right. We are talking in terms of what? Watts, microwatts. Yeah, those thermocouples, they measure nothing but power, but power is directly what? A measure of microcal per second. That's your power, right. So then if I don't do anything, then I will get what? A very flat line, right. Now depending upon whether the energy is released into the system, which is my exothermic process, or taken from the system, the endothermic process, I will get either the downward peak or the upward peak. By convention, my exothermic peaks are the downward peaks, right. So if I have something to titrate in, right, it will come down, down, down until it gets, and then it will reach its the final intensity, and then the peak will go up. So I had on purpose drawn this curve very broad, because I have to explain something else. But when you will see in the curve, they are very sharp peaks, right. So you are titrating in something, so there is some differential power what you had measured, right, and then you allow it to equilibrate before you start the next injection. right? In titration, what do? You keep on adding the titer, right? Right? So then the next injection will come again. Yeah, will go again and flatten out, right? Now the question to you is, if I ask you, this area is what? This area is what? Yeah, enthalpy comes later. It is nothing but the power what you had measured, the microcal per second, or the total heat what you had measured, right? So area under this curve, right? Then you keep on doing the multiple injections, so it will go like this, then up, then like this and up, then like this and up, like this and up, like this and up, and at some point of time it will saturate your entire system, right? Now what I can do, I can take the tip of every peak, right, and I can kind of join them. Yeah, maybe it goes something like this. So some kind of tailing effect what you will see once it saturates, right? So it becomes quite flat in that sense, right? So if you would see, you get kind of sigmoidal curve. Okay. Now when it comes to this microcal per second, the power, it's nothing but, as you rightly pointed out, it's nothing but enthalpy. And I know what concentration of ligand I am titrating in, and I already know what concentration of protein I am using in. So can I also not know the molar ratio at every injection, right? So you actually replace, you will get the same profile. Instead, if you change this to enthalpy versus molar ratio, right? So to begin with, you get this raw data, which is power against time, right, which also turns out, which also after some processing of course turns out to give you a curve with enthalpy against molar ratio, right. This makes more sense, right. And what I can do, instead of saying the area under the peak, I can just have these dots. So what you will also see is this kind of curve, like that. So every dot is nothing but the area under the peak what you have got. Okay. Now once all of it is done, what do I need to do? I need to fit to these points, right. I have got some points, right. Right now they are not connected, right. Right now they are not connected. So let's show them not connected, distinct points, because every point is nothing but the area under that particular curve. Or let's say you have points like these, right. Now what kind of fitting is required? So there are different model systems, right, which defines the stoichiometry, whether it is 1:1 interaction, whether it is 1:2 interaction, or whether it is 1:many interaction. So you have models for all of them, right. Let's say if I am talking of say H2O, so my stoichiometry will be 2:1 for hydrogen is to oxygen. But for proteins, if it is ligand and you have only one active site, then it is probably 1:1 interaction, right. But let's say if it is like a dimer of a protein, then if you are considering one protein, right, as a monomeric, and one monomer is having two active sites, right, not a dimer, but let's say two active sites in it, so then the stoichiometry will be 1:2, right. Or then at times you have this like actin which I work upon. So, it is quite an aggregated, and when you try to disrupt this actin with various drugs, so then you will say one aggregate particle binds to multiple molecules of ligands, right. So, it is to multi-site binding, thus you call it as 1:many. Okay. So, you select any of the model and then hopefully you get a good fit like this. So, different models you can use, and then this helps you to understand the stoichiometry, right. Then again this also gives you the KD value, right. And then if I have all the areas being added up, what will I get? Delta H dot. Will I not get that? Remember I wrote an equation, and from this if you do the relevant subtraction, you will end up with, let us say, delta H protein ligand, that kind of scenario, right, the real one. Now, this you can do at normal temperature and pressure to get delta H naught, the standard conditions, the delta H naught. So, delta G at equilibrium you mean. So, then you will also get like delta S naught information from here, the entropy information, right, at equilibrium. So, that is an indirect measurement of entropy, not the direct measurement. What you measure directly from a ITC is delta H, right, delta H. Okay. So, this is no. So, for every particular model you have these different equations which help you calculate KD, right. So, it is not the standard Michaelis-Menten kinetics what you will get, maybe for 1:1 maybe yes, but then 1:many, then you have this complex equation which helps you calculate this KD values. But that KD value can be calculated only when you do the plot versus when the heat versus molar ratio, right, not the raw data. Microcal per second versus time, you cannot do that. Now, the practical problem I can example I can give you, if I am wanting to work with say Ayurvedic concoction, not the Ayurvedic isolate, but say Ayurvedic concoction, let us say anything like Brahmi for instance, if you want to say. Now, Brahmi is not one component, like there are many components in Brahmi, but you really do not know the concentration of one particular active component over there. Then if you do not know that, then how do you get the molar ratio? You cannot, right? You cannot get the molar ratio. Let us say a Brahmi has 5 components, I am calling it as C1, C2, C3, C4 and C5, and out of this from your work you know that C1 is the active one, right. Now, C2 to C5 they are there, but then for the total concentration they also contribute, but how do you know what fraction is C1? So, that leads to the problem of calculating a molar ratio, right. So, you might be able to, not might, you will be able to get the raw data for the concoction, but then you will not be able to fit it in a proper system, because you do not know what concentration of C1 the active component is into the system, right. Fine. So, is these kind of things clear? So, what ITC measures and how it translates into the different parameters what we want to compute. Now, how it is relevant to us for CMC? Can anyone come up with idea? So, we want to do the CMC, right. So, you expect that up to CMC the enthalpy change is different, then you see a difference and then it saturates. So you expect to have some kind of transition point for the detergent, right? So now you definitely will be doing the 3 detergents, right? So now Mayuresh is going to help you with the experiments, right? Is he around? Yeah, Mayuresh, can you please come around? Right, so Mayuresh will help you, but for this video purpose, right? So for this video purpose, we will be doing only one detergent, that is the SDS. SDS or CTAP? We will be doing the CTAP, which is like the cationic detergent what we will do, right? And then the same thing we are going to do for SDS, and we are going to do the same thing for NP40, right? Just to keep the video short, we will be doing only CTAP for today, right? And of course, I have to introduce Rupesh. You have to turn around the camera, or maybe you can come here on this side without doing anything. So he is our camera specialist, cameraman. Because of pandemic, he has changed his profession, right? So now we can stop here, and then we will resume once we go to the ITC instrument, right? Okay, thank you. Right, so this is what is an ITC machine. It's a Malvern ITC 200, yeah, machine what we have got. It's a Microcal ITC 200, and we are going to do the all the titrations using this piece of machine. Right, now I will hand it over to Mayuresh to explain you how to do these things.
Okay.
Does this lamp work?
Yes, sir.
Yeah, so we can use this light also while we are showing them what's happening, right?
Okay,
so you can turn it off for now.
So
Yeah, so Rupesh, you might have to come back and forth to give a good view. Yes.
So students, this is the ITC 200 instrument. It is called as 200 because the sample volume what it takes is 200 microliter, 200 to 250 microliter. So this port is for our titration and reference. As sir has explained in the earlier part, you can see inside there are 2 ports. I don't know it will be visualized or no.
You have to go up, maybe. You have to zoom in a bit.
So you can see 2 black spots. So the one in the center is our titration port, and the one in the right side is the reference port, where we will be putting our buffer or a solvent in which chemical like macromolecules or the substances which we have to titrate are dissolved. So the center one is for titration 1. The titrant which we will be using for titration is filled in this syringe. You can see. I will just lift it up.
Yeah.
Can
you turn on the light so that they can see it better?
So this is the rest position of the syringe. Here you can say hold or fill the syringe, you can say. So you can see the needle, it is a very fine needle. So whatever titrant you will be using should be a water soluble or a buffer soluble. There should be no particles or the syringe may get blocked. So the solution should be a totally transparent solution. There should be no particles in it.
Okay. That's a Hamilton syringe, right?
Yes, Hamilton syringe.
Yes.
So I will.
What's the capacity of the syringe?
The total capacity is around 40 microliter.
Right.
So it will be shown like how much microliter the syringe has taken from the solution.
Right.
So I will just keep it in again in the clean and dry position till we are not using it, you can say. Then I will show you from the start the software of the instrument.
Can you close everything and show from the beginning? Yeah.
Okay. So this is, you can see here the Microcal ITC 200. This is the software of the instrument. So just double click on it. So see here, pipette success. Okay. let it allow for some time to connect with the instrument. So it's still processing. Yes, now it's done. So you have to go to systems and we have to link the instrument with the origin. So whatever data you will be obtaining will be in the origin format. So this is the origin format. So you can see a graph which was, I think so, done last time. What you can say, the last experiment on the instrument, it is visualized. So this is the main software of the instrument, you can say, where you have to put your parameters or how to handle an instrument, everything like washing, cleaning, filling, all the things can be done with this software. So you can see this is the advanced experimental design. So in this you have to set the parameters like how to you have to conduct the experiment, whatever experiment you have to do, like how much injections you have to inject. So there will be, you can do a 40 injections also depending upon the volume what we will be using for injecting. See for like, since we are using 2 microliter as our injecting volume, we can go till 19 injections. But
19, so 2 microliter at a time, so per injection, right?
Yes.
Okay.
So if you are using 1 microliter as a sample volume, then you can go to 39 or 40 injections also, like that. Then there is the cell temperature, the thermostat what we were telling you about. You can so this can go up to 80 degrees Celsius till from you can say 10 to 80 degrees Celsius. So at whatever temperature you want, you can conduct your experiment like that. Then this is the reference power. The reference power is like the comparison between your titration cell and the reference cell. There should be not much any difference in the volume, so that the energy what is supplied to both of them should be not we can say much more difference than what we have set. So we have set it at 10, so it should be somewhere around 10 or like that. Then comes the initial delay. Initial delay is like we have to equilibrate both the solutions. So once the solutions has been equilibrated, we will give a 120 seconds of initial delay, and then we can go with our injection titration. Then we have the syringe concentration. Syringe concentration in millimolars. So we have to put the concentration what we will be using as a titrant in millimolar, then cell concentration, and then stirring speed. Stirring speed should be 500 or whatever you want, like 250, 300, whatever. Then the data file name. Okay, we have to give the name. Next comes the instrumentation control. So you can see this, the thermostat control. So here you also have to set at what temperature you have to conduct your experiment, like 30 degrees, 37, 45, and then click on the set jacket temperature. So it will set the temperature at that particular temperature. Then these options are like for manual handling of the instrument. If the instrument is not working properly, then you can what you can say operate the instrument manually also. Then this is the what you can say washing or the working modes for syringe. So here you have cell and syringe wash, syringe fill. Syringe fill means titrant filling. Then cell buffer rinse, cell water rinse, detergent soak and rinse. Detergent soak and rinse is done when the syringe is not absorbing the what you can say the titrant properly, or not injecting the titrant properly,
not sucking up.
Yes, and then even the cell, they, like means you are not getting a proper data of the known samples also. Then you have to do this part so that everything is cleaned properly, and then you have to use for the instrument. Then syringe wash, syringe short wash and long wash, then dry syringe. These are the modes for them. Then here comes the real time plot. So now you can see that, as you have seen, the thermostat is at 25. So now the temperature inside the port is around 25.126, like that, and the difference between the two is 0.2 microcals per second. Then comes the setup.
Now go back to the previous screen. So the
Yes.
difference between the reference and the titration cell, right?
Yes, titration cell is
correct.
around
So-
0.2.
Sure.
Then comes the saving, the whatever in which of the folder you want to select and all, like that. And here is the y-axis also. You can change how much y-axis you want, like millicals per minute, microcals per second, microwatt per something, like that. Okay, so now we will go to instrument control. We will be starting with the syringe filling. We will be filling the syringe with the titrant.
Yeah. So you already had cleaned the syringe and kept it ready.
Yes.
Yes.
So I have already cleaned the syringe. So 1 minute. So this is our titrant for the first experiment, C-type, which is of 15 millimolar. So I will be taking in a small Appendorf.
And while you are putting in an Appendorf, please make sure that there is no frothing.
Yes, there should be no air bubble inside it, so that the syringe will not absorb the titrant properly. So I will be just putting it at the load position. This is called the load position, where the syringe absorbs the titrant. So now,
Yeah, it sucks up the titrant here, right?
so I will click on the syringe fill option.
Wait.
So it will show you, like, place the pipette in the rest position. So as I have shown earlier.
So now we can turn on the light so that we can show it moving here.
Yes. So gently hold the syringe with both the hands, because this syringe is very costly. It comes around 2 to 2.5 lakhs, only the syringe. Okay, so this is the rest position of the pipette. So once we are kept there, there is then what you can say, assembly we have to form. I will just show you here only how to do it. So initially the syringe will be somewhat like this. Okay, so first thing is to lose this silver, what you can say, knob. Then pinch this, what you can say, tubing inside the port, and finger tight the tubing, and again slowly tight the silver knob down, whatever we have loosened. Okay, so this is the, what you can say, assembly we have to form. Now we have to put the pipette in the titrant. You can see from bottom the pipette is inside the titrant.
The tip of the syringe is in the solution, right?
Yes.
Yeah, and then you give the command OK.
Okay, now you can see there will be filling of the syringe. So you can see a white valve moving up and down. So that is
the plunger,
you can say, which is sucking the solution from the pipette. Yes, you can see now that white plunger. So now it is mixing the solution. So it 2 times it will suck and release the solution mix in the append of again, and third time it will be sucking the last time.
Yeah.
Like
so
that.
This helps them to avoid any air bubble, right?
Yes.
Getting sucked
And even the homogeneous mixing of the solution.
Yeah. Yeah.
Done.
It's done.
So now it says us, make sure the cells are filled and please disconnect the syringe tubing and insert the pipette into the cell port. So now we have not filled our cell port yet. So what we will do, we will just release the assembly and put the pipette at the clean and dry position. So I will just say OK. Again keep the pipette at the rest position. Whatever you have to do with the pipette, do at the rest position. So again I have loosened the silver knob and tubing, and I will keep the pipette at the clean and dry position and place the tubing here. So this is the position for the pressure tubing. This we are done. Now we will be filling our cell port. So students, I have taken the milky water what we have used to dissolve the SDS, C-tab, twin, whatever we will be using. So now I will be filling the reference buffer.
The reference cell, right?
Reference cell. So what we will be doing, we will be just filling the port at till the mark of the black spot, you can say. So students, you can see I have filled just the reference cell port, you can say, and the one in the middle is not filled yet. So you can see the difference between the two. The right one is just filled while the in the center is not filled. So I have just filled till the mark of the you can say the opening of the port.
Till the brim.
Till
right?
the brim.
Yeah, till the recommended capacity of that cell.
Okay, so now I have also filled the titration port or the cell port, you can say. Now you can see both the ports are filled with the solution till the brim. You can see, yes, it's shining at the brim position. So we have filled the cell port. Now what we will be doing, we will be putting this pipette inside the cell port.
Syringe,
in the
right?
Syringe in the cell port, in the center one. Yes, I will just put it slowly down. Yes, so this is the last position, it will go down. Okay, done. So now we will be starting with in the software thing. Yes, so we will be doing this at room temperature. So we have kept the thermostat at 25 degrees Celsius. We will be putting the syringe concentration, like the C-tab concentration, as 15 millimolar. Okay, and this we will be keeping as 1000, and we will be labeling as
So you had mentioned the concentration of the
Yes.
titrant.
We can change the concentration of the titrant later on also.
Right.
There is not a issue, but we can put it. It will be better if we put now. So we can put as H tab only, and I will just see everything is okay.
Yeah.
Thermostat. So you can see
Now,
the
just sec. It is
Yes.
very important to put in these concentrations because these concentrations are required for that molar ratio to be calculated.
yes.
Yeah.
So you can see now here the instrument is showing the thermostatic ITC at 25 degree Celsius. So then, okay, done. So what I will be doing, I will all the things are okay now. We will just click on the start button here. Okay. So you can see now it will be equilibrating both the solutions in both the ports
That's the baseline.
to form the baseline.
Yeah, that's the baseline formation, right?
And you can see here the
The stirring has started,
part.
right?
Yes.
The stirring has started, so it
So
goes
what we
round
have-
and round over there.
So what we have set for 500, now it is spinning at 500 rpm. Like, so this is the stirring part, so that the cell port is properly homogenized and the injection will be properly monitored. So for forming the baseline, it takes a time. So it takes around 6 to 8 minutes or maybe 10 minutes for equilibrating.
Yeah. So then we can pause here and then we'll come back to the next stage.
So students, you can see it is the solutions has been equilibrated, but it is still the instrument is still what we can say equilibrating. So at any point the system will notice that the equilibration is done. You can see a very flat line. So this is where you can say the solutions are equilibrated. So anytime the instrument will notice it and start our experiment. So we will just wait for that moment. For that moment. Okay, so students, our equilibration is done. So you can see here ITC pre-titration delay. So what we have put our as an initial delay of 120 seconds, it has been started. So now the instrument will take 120 seconds of a delay, and you can say the instrument will start injecting our titrant. So you can see in the right corner here, it is showing that our syringe has taken 39.14 microliter of the titrant, the CTAP solution in the syringe. And you can monitor the what you can say the experiment time also. You can see here time left is around 39 minutes now. So the total experiment will be taking 39 minutes to complete the all 19 injections.
So what we will show them are the first 5 injections, and then we will tell them like how the data looks like after all the 19 injections are done.
Yes. You can see it.
How many seconds done?
So around 80 seconds are done.
40 more seconds to go.
Yes. So that time we can see our first injection. So above just my marker, the the line is running where the baseline has formed. So we have our first injection. You can see. So after the first injection, you can see there is some spike, and now it is coming back to its baseline what we have set, as it was shown in the earlier sir's explanation part. So it will come back to its baseline, and after 120 seconds it will be injecting the second injection. So it has come to our baseline.
Yeah, the baseline has to stabilize before it does the second injection, because you don't want the overlapping areas of the two peaks.
So now it has passed around 230 seconds.
Mm-hmm.
So another 10 seconds there will be another injection.
Yeah. So 120 plus 120, 240. So at 240 you should see the second injection.
And the next injection is right now.
It's here.
So it will be a what you can say, a major change in the heat, because this injection has a volume of 2 microliter, a larger volume, but it will still comes to its baseline.
Yeah.
So I will be showing the second injection when it comes to the baseline.
Yeah.
You can see the difference.
No, you must be wondering why the first injection is the small one, because the first injection is not exact 2 microliter.
Yes.
It is slightly smaller volume what it injects in the first cycle.
Yes.
From the second cycle it becomes optimized and injects exactly the 2 microliter.
So in the first injection it was only 0.4 microliter.
Yeah.
So I will just show you the difference now. So now you can see yes. So you can see which first injection is this much, and the second injection of 2 microliter.
Yeah. So that's
Yeah.
true, because this is like almost 5 times the first peak, right? The second peak is almost the 5 times the first peak. So the first peak it is 0.4 microliters, and the second peak it is exactly 2 microliters injected.
Yes. So in the similar way, the third and the other injections will go on. So we will be showing you till 5 injections.
5 injections.
So in the another 8, 7, 6, 5, 4, the next injection will go. Yes, so you can see. So after every injection, the solution will get saturated inside the cell port, so there will be a decline in the graph. So after every injection there will be a decline in the peak values. If you compare, you can see there is a slight decrease in each of the reactions, reaction peaks. You can see a very slight decline. So once it has reached its micelle concentration, there will be a total decline, and you will may get a very small peak of the graphs. So this is our fourth injection. I can show you the time left also. So it will be 30 minutes more for the experiment to be finished. So this is, so I'll be just showing you the initial 5 injections, and the later data I will be showing after the completion of the reaction or the experiment. So in another 2 seconds, one more injection. Yes.
Right, so that's injection number 5. Now this is we are going to have injection number 10. How much time is remaining? Oh, it's the total time what it tells you, huh?
So it will be
It
taking
should be at
120 seconds.
1200 seconds, right?
Around, I think so now.
Yeah.
Yes.
Yes, so there you go. This is injection number 10, and then we will also show them injection number 15 and the last injection number 19.
19.
So we are almost there. So it is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and the 13th is going on, right? And it also tells you here that you are doing injection number 13, right? So we had showed you like 5 injections, then the 10th injection and the 13th injection is going on right now. Likewise, things is going to, now you more or less have an idea how up to 19 it will go, because this is the kind of data what you will get. Now we will show you how the complete after 19 injection, how the data would look like. Can you go to the
Yes.
analysis screen data? Yeah. So show them how do you import the data. Start from the scratch, please.
Okay. So there is another software from Melbourne, you can say, this one which is called as microcal analysis. So in this you will be analyzing your ITC data. So we have to select your instrument like ITC 200 what we are using now. Then there is this option select data, first option. So now you will be selecting the file. You have to what you can say, export your
The raw data over
the
here,
raw
right?
data, you can say.
Yeah. Can you go back to that curve? Those peaks, sorry, not the curve.
So this one.
No, not this one. The yeah. So,
Yeah.
can you zoom in a bit? No. On the screen, can you zoom in a bit? So let the camera stay here. Right. So show them the first 5 injections on this.
Yes. So you can see this is this was our first injection, then second, third, fourth, fifth, and so on.
Up to 19.
Up to 19 injections.
Yes.
So this is the earlier one what we have done.
Right.
So this is your titration data.
Now here you are just titrating in CTAB into water, and your reference cell is already a water, so you don't have to do any buffer subtraction, right? So that's already the taken care of data. Can you show them all the peaks, all the 19 peaks? How do they look put together? Yeah. See, this is what it is. So all the 19 peaks are here. So after 19 injections, so it has kind of saturated.
Yes. So in these we have to do our analysis. So if you have not put your concentrations properly, you can change your concentrations from here. Then there is another option of removing the bad data. So we will be removing this point, our first injection of 0.4 microliter, so that it doesn't interfere with our analysis. So just double click it and it is gone.
Now we have got the data here. So and we had done the fitting, the curve fitting for it. Now for this particular experiment, there is no one side, two side or multi-side binding required. All it's required, like you have this smooth data, you draw tangent here, tangent there, and then draw the transversal, then the half midpoint will give you the inflection point where it changes from the monomeric detergent to the micelle, right? This is our whole idea, that this is like a monomeric detergent to micelle formation. Do we see this inflection, right? So this is what is required, and this likewise the data is available for you for doing things for SDS and also NP80, right? So now it's up to you to analyze the given raw data and get us back with your reports depicting the CMC values for the 3 detergents, right? CTAB, SDS and NP80. Okay, so I'll sign off for now. Thank you very much. Goodbye and take care.